Question number 1. The medium used in membrane filter technique was option A EMB agar, option B EMR-VP medium, option C lactose broth, option D endo agar. This is the most basic question. So the correct answer will be option number B EMR-VP medium. And EMR agar, EMR VP medium, lactose broth. So EMB agar क्या है? एक differential medium है. EMR VP medium it's also a differential medium, whereas lactose broth ये एक broth medium है and endo agar it's a differential medium. Next, Lysol is a option A sterilant, option B disinfectant, option C antiseptic, option D antifungal. So Lysol it is an disinfectant. My correct option will be option number B. Actually disinfectant क्या होते हैं? Disinfectant is a chemical agent that is used to destroy or eliminate microorganisms on the surface of the object. मतलब कोई भी objects के surface में कोई microorganisms होगा तो वो उसको eliminate या destroy करता है. Whereas antiseptics क्या होते हैं? Antiseptics is a substance that is applied on the living tissues to reduce the risks of inspections or sepsis or putrefactions. Whereas antifungal agents क्या होते हैं? इस एक टाइप के medications होते हैं और chemical that is used to treat the fungal infections. So Lysol it is a disinfectant. Question number 3. Which of the following is a neutral stain? Option A picric acid, option B GEMSA, option C neutral red, option D malachite green. So what exactly neutral stain क्या होते हैं? Neutral stains are those stains that do not have the affinity for either acidic or the basic components of the cell. उनके पास कोई भी affinity नहीं होता towards acidic or basic components of the cell. Picric acid actually it's a yellow crystalline solid which is used as a fixative. So picric acid it is used as a fixative. Whereas option number B GEMSA. So GEMSA क्या है? It's a differential stain which is used to differentiate between different types of microorganisms. Option number D malachite green. Malachite green क्या है? It's a basic stain which is used to stain spores or vegetative cells of the bacteria. So neutral stain क्या होगा? Actually neutral stain it's neutral red. That is option number C. Question number 4. Peptone water medium is an example for option A synthetic medium, option B semisynthetic medium, option C differential medium, option D none of these. So peptone water medium it's an example for the semisynthetic medium. Actually synthetic mediums क्या होते हैं? Those mediums which is defined as a medium that contains only pure ingredients whose exact composition is known. So these are synthetic medium, whereas semisynthetic mediums जो होते हैं वो जो medium that contains both chemically defined and complex components. These are semisynthetic mediums. And differential mediums को कैसे हम define करते हैं? As a type of medium that allows both, means that allows the growth of multiple microorganisms, but it distinguishes them on the basis of their biochemical properties. So इसमें हमारा correct option number B है. That is semisynthetic medium. Peptone water medium is an example of semisynthetic medium. Question number 5. The method in which the cells are frozen dehydrated is called option A pasteurization, option B desiccation, option C in disinfection, option D lyophilization. So methods in which the cells are frozen dehydrated. So frozen dehydrated, actually lyophilization ऑप्शन डी हमारा राइट भी आंसर है। सो लाइफलाइजेशन एक्चुअली होता क्या है, इसको हम लोग फ्रीज ड्राइंग भी बोलते हैं। लाइफलाइजेशन इज ऑल्सो नोन एज फ्रीज ड्राइंग। सो इसमें क्या होता है, लो टेम्परेचर डिहाइड्रेशन प्रोसेस होता है which involves the freezing products, lowering pressure that removing the ice by sublimation process. So the method in which cells are frozen dehydrated is called lyophilization. Next, question number 6. The technique used to avoid all or microorganisms is accomplished by option A sterilization, option B disinfection, option C surgical sterilization, option D disinfection sterilization. So the technique used to avoid all microorganisms is accomplished by option number A, that is sterilization. So sterilization क्या होता है, just हमने अभी समझा कि sterilization is a process of destroying all living organisms and viruses. It is used to eliminate or kill all the forms of microbial lives, which includes endospores, bacterial endospores. Okay, and disinfections क्या होता है, disinfections they actually it's not necessary that they kill all the microorganisms, but it reduces the number to an acceptable limit. मतलब उसके बाद एक लेवल तक उन लोग उसे eliminate करेंगे and that it does not harm the human health. So my correct option will be option number A, that is sterilization. Question number 7. Thermal death time is time required to kill all cells at a given temperature. Option B, temperature that kills all cells in a given time. Option C, time and temperature needed to kill all cells. Option D, all of the above. Actually thermal death time हम इसको ऐसे T D T भी बोलते हैं, it's short form में TDT. So thermal death time, it is termed used to describe the time which is required to kill all the vegetative cells of microorganisms in a sample at a given temperature. So ये TDT, that is thermal death time, is used to describe the effectiveness of sterilization process that is used to high heat, that is example autoclaving के time. So my correct option will be option number B, that is that is the thermal death time is a temperature that kills all cells in a given time. Question number 8. A culture medium, the exact composition of which is not known. Means the composition of which is not known. Option A, simple. Option B, complex. Option C, defined. Option D, natural. So what exactly complex mediums are? Complex mediums होते हैं culture mediums that contains only few components, whereas complex mediums होते हैं a culture medium in which the exact chemical composition of each components of the constituents is not known with certainty. So यहाँ पे हमें question में पूछा गया of which it is not known. So my option will be option number B. और हम और C भी देख लेते हैं एक बार. Defined it is termed as a culture medium in which the chemical composition of each of the constituents is known with its certainty. And option number D, that is natural medium. So as the name is suggesting, natural means it is not artificial, but it is that it is not artificially prepared and it is obtained from the natural sources. My correct option is option number B, that is complex medium. Next question. Number 9. A Lux chill diffusion test is used for the detection of option number A, tetany toxin. Option B, cholera toxin. Option C, diphtheria toxin. Option D, toxoid. So Alux gel diffusion test ये क्या होता है, is used to determine whether or not a stain of Corynebacterium diphtheria is toxigenic or not. So positive या negative determine करता है. So my option will be option number C, that is diphtheria toxin. Question number 10. The temperature required for pasteurization is above 150 degrees Celsius, option B 100 degrees Celsius, option C 110 degrees Celsius, option D none of these. So pasteurization में हम mainly two types पढ़ते हैं, that is LT low temperature long time and HTST that is high temperature short time. So LT में क्या होता है, it involves heating of milk to 65 degrees Celsius for 30 minutes, whereas HTST process में होता है, it requires heating of milk to 72 degrees Celsius or to 74 degrees Celsius for 15 to 20 minutes. That means the temperatures are all below 100 degrees Celsius. So my correct answer will be below 100 degrees Celsius. Question number 11. Separation of a single colony, bacterial colony, is called option A isolation, option B separation, option C pure culture, option D all of these. Again an easy question. So separation of a single bacterial colony, it is known as isolation. That is option number A. Isolation क्या है? This is a process which is used to obtain a pure culture of bacteria from the mixed culture. Question number 12. Which of the following is ionizing radiation? Option A UV rays, option B IR, option C gamma rays, option D none of these. So which of these is ionizing radiation? So ionizing radiations क्या होते हैं? It's a type of radiation that has enough energy to ionize atoms or molecules by detaching electrons from them. So this ionizing radiations can be gamma radiations or nuclear radiations, X-rays, cosmic rays. So my correct option will be option number C, that is gamma radiation. Which of the following question number 13, which of the following induces dimerization of thymine? So it is option number B, that is UV rays. UV rays induces dimerization of thymine. Question number 14. When food materials are preserved at a temperature just above freezing temperature. So यहाँ पे ध्यान दीजिए, just above freezing temperature, the process is called freezing, option B pasteurization, option C chilling, option D frosting. So freezing क्या है? It's a method of preserving food by reducing its temperature below its freezing point. So ये right answer नहीं हो सकता, since this process is used for long-term preservation of food items. Pasteurization, इसमें heat use होता है. Option C chilling. So chilling, it's actually the correct answer also. So when food materials are preserved at temperature just above freezing temperature, so the process is known as chilling. Whereas frosting क्या होता है? Frosting actually is termed, this term is used for means when the formation of ice crystals on the surface of food items. जब भी food items में ice crystal का formation होता है, when the temperature of food item is below the freezing point of the water. So उसमें जब ice crystals का formation होता है, जब हम उसको term करते हैं as frosting. So here my correct answer will be option number C, that is. Question number 15. Which of the following method of sterilization has no effect on spores? Option A drying, option B hot air oven, option C autoclave, option D none of these. So actually option A is correct option, that is drying. So drying क्या होता है? It's a method of sterilization in which the process of removing microorganisms by applying moisture free heat. Okay, it is based on the principle of conduction. And when the heat is absorbed actually by the outer surface of the food item, and then it's passed towards the next layer. So पहले वो outer surface में apply होता है, then it passes to the next layer. So my correct answer is try. Question number 16. If the vectors transmit the infection mechanically, they are called option A biological vectors, option B mechanical vectors, option C biological reservoir, option D both A and C. So if the vector transmit the infections mechanically, they are known as mechanical vectors. So mechanical vectors क्या होते हैं? Actually, vectors are animals that they carry a pathogen from one host to another without being infected themselves. मतलब वो host वो खुद infect नहीं होते हैं, whereas वो जब किसी और के ऊपर बैठते हैं, they infect them. So mechanical transmission, so the means actually the pathogens होते हैं, this pathogen is carried on the surface of the vector's body and it is transferred to the host when the vector feeds on it. Example, flies. वो जब कोई contaminated food कोई contaminate चीज के ऊपर बैठती है and then it goes and flies, और वो एक food material के ऊपर बैठ जाती है, food item के ऊपर, और उस food item को human consume कर लेता है. That means it's a mechanical vector. Whereas biological क्या होता है? Biological vectors, actually biological transmissions, it occurs when the pathogen it multiplies within the vector's body and is transmitted to the host through the vector's saliva or the feces. So here my correct option will be mechanical vectors. Question number 17. Autoclaving is carried at dry heat, atmospheric pressure, option C 120 degrees Celsius, option A or D all of these. So autoclaving क्या है? It's a sterilization process that uses high pressure steam to kill the microorganisms. It even kills the spores. So the heat, the temperature which is used in the autoclaving, it is 121 degrees Celsius, that is 250 degree Fahrenheit for 15 to 20 minutes. So my correct option will be option number C, that is 120 degrees Celsius. Next, question number 18. Temperature in pasteurization is option A 62.8 degrees Celsius, option B 35.7 degrees Celsius, option C 68.2 degrees Celsius, option D 60.8 degrees Celsius. So just few slides पहले हमने पढ़ा pasteurizations के two types, LT and HTST. That is low temperature long time and high temperature short time. So इसमें क्या होता है? LT method में क्या होता है? Involves heating of milk at 65 degrees Celsius for 30 minutes, whereas HTST process it involves heating of milk to 72 degrees Celsius, 72 to 74 degrees Celsius for 15 to 20 seconds. So here mainly क्या process के बारे में बात किया है? LT, that is low temperature long time के ऊपर. So that is option number A, that is 62.8 degree Celsius. Question number 19. The bacterial culture prepared by pure culture method is option A inoculum, option B suspension, option C dilution, option D none of these. So the bacterial culture which is prepared by pure culture method, actually pure culture क्या होता है? That contains only one type of microorganisms. Mainly pure culture में हम लोग streak plate method use करते हैं, which involves spreading the inoculum back and forth across the surface of the agar plate to physically separate the microbial population. Then suspensions actually क्या होते हैं? Suspensions एक heterogeneous mixture है in which the solid particles are spread throughout the liquid without dissolving it. And dilution means इसको dilute करना means the process of reducing the concentration of a solute means solute का concentration को reduce करना. A solution is that is actually a solution is added more by adding more solvent. So my correct option will be option number A, that is inoculum. Question number 20. Algae are rich in carbohydrates, proteins, vitamins, all of these. Algae, they are rich in several nutrients, जिसमें omega 3, omega 6, omega 7 fatty acids पाया जाता है, vitamin B12, C, E, minerals like calcium, potassium, wide range में carotenoids पाया जाता है. So they are rich in all. Question number D, all of these. Question number 21. Lysine is produced from option A, Corynebacterium glutamicum, option B, Clostridium botulinum, option C, Mycobacterium species, option D, Pseudomonas. So lysine actually क्या होता है? They are mainly produced by submerged fermentation, mainly by the Corynebacterium glutaminicum and Corynebacterium glutamicum and E. coli, that is Escherichia coli. ये क्या होते हैं? 2 types of fermentation methods होते हैं, वो industrially इसको produce करते हैं lysines. One is indirect fermentation and another is direct fermentation. Question number 22. The orderly increase in the quantity of all the cellular components is known as option A, reproduction, option B, growth, option C, binary fission, option D, none of these. So orderly increase बोला है. Name is suggesting orderly increase in the quantity of all the cellular components. That will be option number B, that is growth. Reproduction क्या होता है? In which organisms they make a copy of themselves and provide for their survivals. Whereas binary fissions क्या होते हैं? Binary fissions, it's a type of asexual reproductions which is mainly it is seen in bacteria. A single cell that means a single cell organisms, it splits into 2 individual organisms. Question number 23. Thiobacillus thiooxidans, they grow at a pH of 7, 1, 6 or 9.5. So Thiobacillus thiooxidans, they grow at a pH of 1. Actually Thiobacillus thiooxidans क्या है? It's a gram negative rod shaped bacteria है जो that uses sulfur as its primary energy source, and it grows at a pH of 1, and it is actually an acidophile, aerobic and a chemolithotroph microorganism. Question number 24. Slow freezing requires the condition 0 degree to 15 degrees Celsius for 15 minutes. Option B, -6 degree to -10 degrees Celsius for 10 minutes. Option C, -15 degrees Celsius for 3 to 72 hours. Option D, none of these. So slow freezing requires -15 degree Celsius for 3 to 72 hours. Option number C. Question number 25. Discontinuous heating is called option A, pasteurization, option B, sterilization, option C, fermentation, option D, tyndallization. So answer to this question, please आप इस question का answer comment down कीजिएगा. Even comment down your scores out of 25 which you have achieved, and at the end आपको कोई भी क्वेश्चन पे प्रॉब्लम हो, डाउट हो, सो प्लीज डू कमेंट डाउन. नॉट फॉर दिस, इवन इफ आपके क्लास 1 से लेकर क्लास 3 में भी कोई भी डाउट्स रहे, सो प्लीज